In this video, we're going to talk about DNA. DNA is deoxyribonucleic acid. I don't know if I'm going to fit it right there, so I'm going to write it up here. So it is deoxyribonucleic responsible for all of our hereditary information, and we pass that down from our parents to babies. And so it's responsible for transmitting all hereditary information, and it's going to be unique to each individual. And it is a double-stranded alpha helix shape. A nucleotide is a monomer of DNA, and each monomer of the DNA is going to contain phosphoric acid, deoxyribose sugar, and a nitrogen base. The phosphoric acid and the deoxyribose are going to form the backbone, and the hydrogen bonds between the nitrogen bases hold the strands of the helix together. And those nitrogen bases can be different. And here is that the helix, if you will, of DNA loops around each other based on the really strong attractive forces between the nitrogen bases. Let's look at those four DNA bases. Four DNA bases, cytosine, which is given the symbol C, thymine, which is C, thymine, which is given the symbol capital T, and adenine, which is given the symbol find these bases in DNA. So cytosine always bonds to guanine. So what that means is you will have what is called a CG pair. And thymine always bonds to adenine. So you will have the T pair. And the reason this happens is because of those attractive forces on the hydrogen bases. Here is cytosine, and you can see the backbone is phosphoric acid, deoxyribose sugar, and then it's bonded to this nitrogen base. Here we have another nitrogen base, and then we have our sugar and phosphate. And blueprint for protein is going to be three nucleotides that represent a specific amino acid. So the nucleotide sequence for alanine is GCC. That is the code, if you will, in order to trigger your body to make alanine. And a chromosome is a unit of DNA. Pairs of chromosomes, and each pair contains one from each parent. Now we can use all of this to analyze DNA. When we analyze DNA, we use a specific method, and it's referred to as short tandem repeat or STR analysis. So we have short tandem repeat analysis. Eyes patterns of repeating nucleotides in non-coding sequences. So when you hear about people using DNA to identify suspects or identify remains, this is generally what. Let's look at an example of how we can use the information. What percent of the population has the 9-12 pattern on the TPOX and the 10-13 pattern on the D16S39? In order to solve for the probability of having multiple patterns of these repeats, you have to take the first percentage and multiply it the second percentage. If I'm dealing with the 9-12 pattern on TPOX, I'm going to go to the 9-12 and it has a 1.17 probability. So I'm going to take that 1.17%. Remember, percent generally means per 100. And I'm going to multiply that by the 1013. I need to go find the 1013, which is 1.4. So this is going to give me, right, I can do 1.17 divided by 100 and multiply that by 1.9 divided by 100. I get 0.00022. If we want to put that back in a percentage, we can just multiply it 100. So we would get 0.227. So if you were looking for these two patterns together on the same person, there would only be a zero left off at zero here, a 0.0227% chance of that happening. Now, if you're looking for a population size required to find the individual with that matching pattern, you have to take the inverse of the probability. We would take 1 over 0.000227. Notice that the inverse of the probability, not the inverse of the percentage. I can do 1 divided by 0.000227. And that means that I would have to look for 4,000 and people in order to find one person with this type of pattern. Mitochondrial DNA DNA. A lot of sometimes we'll refer to that as nuclear DNA. And then we have mitochondrial DNA. Now, mitochondrial DNA only comes from the DNA of the mother. And there are a lot of benefits to this. The differences between the mitochondrial DNA and the nuclear DNA present is a lot of useful properties in forensic science, but it also has some that are not very useful. Now, there are so many more copies of mitochondrial DNA in each cell. It makes it a lot easier to recover a usable sample, a scarce amount of DNA molecules. So it increases the odds of obtaining a usable sample. First, since mitochondrial DNA only comes from the mother's side, then you cannot distinguish between relatives on the mother's sides of the family. So this limits the forensic science usefulness of mitochondrial DNA when you're trying to identify the remains of a missing person. If you have a set of human remains that is located, but there is no source of DNA for this individual, then the mitochondrial DNA of a sibling mother, a maternal aunt, a maternal grandparent, or a child of the maternal aunt can be compared with the mitochondrial DNA. Are among the missing, then it's really hard to use the mitochondrial DNA.